In this video, we will explore the complex processes that occur during the development of the small intestine. From germ layer formation to the formation of villi and microvilli, we will discuss how the small intestine acquires its functional properties. We will also cover the role of intestinal stem cells, neural crest cells, and smooth muscle development. The script aims to provide a detailed understanding of small intestine development in a clear and accessible manner. During embryonic development, the endodermal germ layer forms the gastrointestinal tract. This process involves gastrulation, morphogenetic movements, and differentiation into specific regions. The midgut region will be discussed in detail. During development, the gut tube undergoes folding and rotation to acquire its coiled shape. The midgut region specifically undergoes counterclockwise rotation and a temporary herniation outside the abdominal cavity. This herniation is due to the rapid expansion of the midgut. Eventually, the midgut returns to the abdominal cavity and undergoes further twisting, leading to the formation of the primary intestinal loop. Proper folding and rotation ensure the correct positioning of structures like mesenteries and blood vessels. During development, the small intestine undergoes a complex process of elongation. Cell proliferation and migration in the intestinal epithelium, coupled with extracellular matrix remodeling and signaling pathways, contribute to its growth. A detailed understanding of these mechanisms is crucial for medical students studying embryology. The small intestine develops finger-like projections called villi through a process called columnarization. Microvilli on the surface of the epithelial cells and the brush border increase the surface area for absorption. Cellular rearrangements and signaling pathways are involved in this process. The development of the small intestine involves the differentiation of specialized cell types, such as enterocytes, goblet cells, Paneth cells, enteroendocrine cells, and Tuft cells. This process is controlled by signaling pathways, including Notch, WNT, BMP, and Hedgehog, which regulate cell fate, proliferation, and maturation. Intestinal stem cells, ISCs, play a crucial role in the development and maintenance of the small intestine. These self-renewing cells reside in specialized compartments known as the crypts of Lieber-Kuhn, located at the base of the intestinal villi. ISCs give rise to transit-amplifying cells that undergo a limited number of divisions before differentiating into the various cell lineages of the intestinal epithelium. The presence of ISCs ensures the continuous replenishment of cells in the intestinal epithelium, essential for tissue homeostasis, repair, and regeneration. Throughout the development of the small intestine, the epithelium and the surrounding mesenchyme communicate and interact, playing vital roles in tissue organization and differentiation. The reciprocal signaling between these two layers involves complex molecular interactions, including the secretion of growth factors, cytokines, and extracellular matrix components. The mesenchyme provides essential cues for the proliferation, differentiation, and patterning of the intestinal epithelium. Additionally, the mesenchyme influences the development of various structures, such as the muscularis externa, submucosa, and blood vessels. Meconium, the first fecal material passed by newborns, plays a vital role in gut maturation. It is composed of various substances accumulated during fetal development, including intestinal epithelial cells, bile pigments, mucus, and vernix caseosa. Meconium formation is not a passive process but rather an active and dynamic process involving the transitional stage from swallowing amniotic fluid to sucking. The presence of meconium in the intestine indicates a well-formed and functioning gastrointestinal tract, ensuring the ability to process nutrients effectively after birth. The development of the mesenteric vasculature is crucial for the fetus's proper nourishment and the small intestine's growth and functionality. Angiogenesis, the formation of new blood vessels, occurs through successive processes, including vasculogenesis, sprouting, remodeling, and maturation. These processes involve the dynamic interplay between endothelial cells, parasites, smooth muscle cells, and growth factors. The mesenteric vessels establish an intricate network that supplies oxygen and nutrients to the developing small intestine, facilitating its growth and functional maturation. In the development of the small intestine, neural crest cells play a vital role in forming and populating the enteric nervous system, ENS, which regulates gastrointestinal function. These migratory cells differentiate into enteric neurons and glial cells, enabling the ENS to control peristalsis, fluid secretion, and environmental sensing. Smooth muscle cells form a key component of the small intestine, regulating its motility and contractile properties. The development and differentiation of smooth muscle cells involve a coordinated interplay of various signaling pathways and transcription factors, including the TGF-beta superfamily, myocardin, and serum response factor. These factors are vital for smooth muscle cell proliferation, migration, and maturation. Once formed, coordinated peristaltic contractions drive the movement of luminal content along the length of the small intestine, facilitating digestion and nutrient absorption. Abnormalities in the development of the small intestine can cause issues in its positioning and functioning. Malrotation, where the midget fails to rotate properly, and intestinal atresia, a blockage in the intestinal lumen, require surgical correction for normal digestion. In summary, the development of the small intestine involves complex cellular and molecular events that establish its structure and function. Understanding this process can lead to insights for treating developmental disorders and small intestine conditions.